Good morning, everyone. Today we're going to look at complex inheritance, as in when do these Punnett squares get a little bit more complicated, and how can we determine the genotypes and phenotypes that are displayed in a population. You're going to want to follow along with our activity here of 4.7 coloring Punnett squares, because I'm going to tell you exactly how to get through some of these, and I'm going to pause the video so that you can work through some of them on your own, as well as check your answers with me here. So it's kind of like a guided tutorial here. So in the first part of 4.7, it's talking about simple inheritance. Simple inheritance is what you've been learning so far on how we just take the dominant allele over the recessive allele, and it'll show up every single time. So in this case, we have rounded ears are dominant over pointy ears. To help us understand the phenotype that is represented, how the dominant allele shows up and recessive allele doesn't show up, we're going to use a yellow highlighter to represent the pointed ears phenotype and a black marker to represent the rounded ears phenotype. So if you need a highlighter, you need a marker, please let me know. I got some right up front. I'll set them out on my table for you. Now, to list out our possible genotypes, for both of these phenotypes, the round and the pointed, we just have to ask ourselves which one is dominant. The dominant one is rounded, therefore all dominant letters would be one genotype, and one dominant, one recessive letter would be another genotype. Here, I chose the letters E for ears, so capital E, and capital E, lowercase e, are our possible genotypes for rounded ears. That leaves only one genotype available for our pointed ears. That would be lowercase e, lowercase e. Hopefully this feels like review. So in number 2 there, you can see that we have a cross between a homozygous dominant parent and a homozygous recessive parent. In the boxes there are places to fill out what are the genotypes that we're crossing. Then we'll put them into the Punnett square and figure out the genotypes that are possible to come out. And then we'll do the same thing. We'll kind of copy and paste it over and color in the boxes to represent round ones and pointed ones. Again, our yellow highlighter is going to represent the pointed ears. The black marker is going to represent rounded ears. And we'll do this in a very specific pattern so that we can cover up the recessive alleles. Okay? Now, if we're crossing a homozygous dominant parent, we'll have a capital E as one. Homozygous recessive parent has lowercase e as the other. We put them into the Punnett square. Now that we have the Punnett square, we're going to drop all the capital E's down this column here, all the capital E's down this column, all lowercase e's. That should leave us with all heterozygotes in this particular Punnett square. And if I put that same thing over here, now I can color in the boxes on the right to represent the allele of the rounded, or the pointed ear. That would be the yellow highlighter. All I'm doing is I'm coloring the boxes that have that lowercase e in it. So it's showing me that the allele is represented there. But then I take my black marker and I color over the yellow marker anywhere there's a dominant allele, which would be all 4 squares. Which means that this actually shows us our phenotype in a different way. We could read the genotypes all day long. This can show us the phenotype right away by separating the genotype from the phenotype and color coding it. This will be very helpful later when we get to very complex ones. So bear with me if you feel like this is super easy. Speaking of the phenotype here, phenotypic ratio here is super easy. All 4 boxes have the black marker. All 4 types are rounded ears, therefore it's going to be 100% rounded ears. If you feel good about that, very good. Move on to the next slide, and I'm going to have to pause it on you to have you try it out for yourself. This next one is not on your paper. I repeat, this one is not on 4.7. It is not number 3. See if you can work it out in your head, and see if you can come back after the pause and see how it worked out. So the homozygous recessive parent and the heterozygous parent are labeled as lowercase e, and capital E. Looks like we have the genotypes listed side by side right here. And if we put that same thing in the color coding squares, then we'll notice that every single box gets a lowercase e allele, and only 2 boxes are going to get the uppercase E allele, covering up that lowercase yellow highlighter. So in this case, with a homozygous recessive and a heterozygous parent, the phenotype ratio would be 2 rounded ears to 2 pointed ears, or 2 to 2 ratio. You could also say 50% to 50%. You could also say 1 to 1, because there's 1 of each. Moving on to number 3. I'll pause it here and let you try it out for yourself. See how you do. All right, things get a little crazy when the heterozygotes get involved here. You can see that a capital E lowercase e and a capital E lowercase e will mix together to give us all of our different kinds of genotypes here. And if I put them in the color coding square, well, we have 3 of them that will be highlighted with yellow because they have the lowercase e allele, and then 3 will be colored with black marker covering up 2 of those yellow highlighters because they have the dominant alleles there. And in this case, it looks like we have a 3 to 1 ratio of black to yellow, which means that would be 3 rounded to 1 pointed. Now, in this next challenge, we're going to be looking at what is called incomplete dominance. Incomplete dominance literally means that the dominant trait is not strong enough to entirely cover up the recessive allele, so we get some sort of mix between the 2 alleles. In this case, we have curly hair being dominant to straight hair. So you would expect that it's either going to be curly or straight hair, but we know that there's hair of all different kinds here in the middle between those 2 different standards. And so we might say that curly hair is incompletely dominant to straight hair, and that wavy hair is that incompletely dominant phenotype, meaning that the middle between curly and straight is wavy. That would be the phenotype in the middle between them. So we're going to use a yellow highlighter to represent curly hair, and this time we're going to use a blue highlighter that represents straight hair. Now that means that the black marker isn't coming into play here, and you'll see why in a second here. Now, in this situation, we have 3 phenotypes here. We have the curly hair, which would represent a capital C, or you could use H's here as well if you wanted to. It makes no difference to me. But capital C for curly hair. If we want straight hair, we have to have the 2 lowercase c's for lowercase c to give us the recessive alleles. And the wavy one would have to be a mix of the 2. So this would be the heterozygote. The heterozygote is no longer going to be only dominant allele showing. It's now going to be a mix of between the 2 of a capital C, lowercase c, almost like it's 50% curly, 50% straight, resulting in wavy. So looking at number 5 here, we have a curly-haired parent mixing with a straight-haired parent. In this case, we know the curly-haired parent would have to have a capital C genotype, and the straight-haired parent would have to have a lowercase c genotype. And that is exactly how we're going to cross them here. Just like normal, we're going to put them into the Punnett square here. I put the curly-haired parent at the top and the straight-haired parent to the left side there. And if we drop all the capital letters down and all the recessive letters through, you should get 4 of the same genotypes here, capital C, lowercase c, in every single box. When we go to figure out how this affects our phenotypes, you could probably see right away that these are all heterozygotes, but in this case the heterozygote is going to be a mix between the 2. To represent that with our color coding, to make it easy on you, you color in with the blue one for all the straight hairs, because all these recessive alleles are in each boxes. And then you color it in with yellow, and yellow plus blue makes green. So now we can visually see, oh, the middle between the 2 is going to be in every single box. The middle between blue and green, or excuse me, blue and yellow is green. Therefore, all these are a mix between blue and yellow. That means that our phenotypic ratio is going to be 100% wavy hair. Give this next one a shot with the 2 wavy-haired parents. This is number 6 in your assignment, so you can do it right there on the paper. Feel free to try it out and see how you do. All right, 2 wavy-haired parents are capital C, lowercase c. Putting them into the Punnett square here, we should get that familiar mix of a 1 to 2 to 1 ratio in genotypes. The capital letters, the heterozygotes, and the lowercase letters. The heterozygous dominant and the heterozygous recessive mixed with the homozygous dominant and the homozygous recessive mixed with the 2 heterozygotes. I get them twisted up sometimes too. Now when coloring this in, I'm going to start with the blue highlighter again. I'm going to color in the lowercase c's right here with that blue highlighter color. Then I'm going to take my yellow highlighter and color in for the curly-haired one, the dominant alleles. That would be this box, and this box. And so that means I get 1 yellow, 1 green, another green, and 1 blue. A 1 to 2 to 1 ratio of all of our phenotypes here to match the genotypic ratio over here. Which means this phenotypic ratio would be 1 curly to 2 wavy to 1 straight. That would be how often the curly hair occurs, the wavy hair occurs, and the straight hair occurs. Give this last one a shot. This is number 7 in your worksheet. So crossing, our straight-haired parent has a homozygous recessive alleles, and a wavy-haired parent has our heterozygote involved. Crossing them here in this Punnett square, you should get 2 of them being heterozygotes and 2 of them being homozygous recessive. And if we color code those, well, every single one of those has a recessive allele in it, so I'm going to color every single one of them blue first. And then to make them yellow, let's see, I've got a dominant one going in that one and a dominant one going in that one. Therefore, only half of them should have our green color, which would be wavy hair, meaning that our phenotypic ratio is going to be a 2 to 2 or a 1 to 1. 50% to 50%, you get the idea of wavy hair to straight hair. There will be no curly hair individuals from this cross. All right, in this next situation, we actually have 2 alleles that are considered co-dominant, which means that they both are very, very strong and will show up. And if they're in the same organism, as in one allele is dominant and the other allele is also dominant, it will not be a mix of the 2, but both will be represented in the same area. So if both alleles are dominant, neither allele will be allowed to cover the other one. Instead, we're going to fill in half of each square with each allele, meaning that it'll be 50% of one allele and 50% another if we have the 2 alleles put together. This is a really good example of one, a sickle-shaped blood for a disease called sickle-shaped anemia is a co-dominant trait where your blood cells have a weird C shape to them, a sickle shape, when they have this S allele attached. And normal shaped cells are going to be labeled as N because normal shaped cells are also dominant. So in order for a person to have only sickle-shaped cells, then they would have to have the 2 S alleles, and these are both capital letters because they're both dominant. In order for someone to have the normal phenotype, we would have to have 2 N alleles. It's weird that we're mixing letters up, but both of them are dominant, so we have to have a new letter to represent a dominant allele. In this case, both capital N's are normal. If we mix the 2, if we have some sickle cells and some normal cells, then that means that the possible genotype there would be an SN, both of them being capital, both of them being dominant. Therefore, this is a co-dominant sequence. So we're going to use a black marker for sickle shape and a blue marker for normal shape. Remember, when we get to ones that are mixed, we're not going to shade both of them all in. Instead, we're going to go 50-50 on them. So in your first example for number 9, your heterozygous parent is going to match up with a normal parent. The genotypes for the heterozygous parent was SN, and the normal parent was a NN. Putting those on the Punnett square, we have SN and NN. The N's are going to travel all the way across into each of the 4 boxes. as well as this N is going to be in these two boxes, making these homozygous for N alleles. They'll be normal. The S is going to drop into each of these boxes, making those alleles SN, and those will be homozygous individuals. Now, what that would look like on our color coding here is that we would have blue as a normal shape be filled in completely for the ones that have the normal genotype, because they'll then have the normal phenotype only. For the ones that are a combination of the codominant genotypes, then that would have to be 50-50. And you can make the line going from bottom left to top right or top left to bottom right. Doesn't matter to me whatsoever. The point of the matter is this is a 50-50 ratio still. It just happens to be 50% of our individuals will have a normal cell shape in their blood, and 50% of the individuals will have both the normal cells and the sickle cells in their blood. By the way, these sickle cells don't work quite as well as the normal blood cells, and therefore don't transfer oxygen as well to different parts of the body. Individuals with sickle cell anemia have a hard time getting oxygen to all the different parts of the body. Give this one a shot. Work through it a little bit slower just so you don't confuse yourself with the 50-50 boxes there. And let's see how you do. This is number 10 on your worksheet, by the way, in case you didn't notice that. When we cross the sickle-shaped parent and the normal parent, we're actually crossing two homozygous parents, and that's going to result in all of our individuals being heterozygous, which means that when we go over here to the color coding, it's actually going to be pretty simple, because you know that this is going to be codominant, so every single box is going to have that half normal, half sickle cell shape in their blood, and therefore it's going to be a phenotypic ratio of 100%, or 1 to, or yeah, just 1, normal and sickle cell mixed here. This last one is number 11 on your worksheet. Let's cross two heterozygous parents. So two heterozygous parents are the S and the N. If we mix those two, we're going to get the 4 different squares, well, the 3 different genotypes in the 4 different squares. The capital S, capital S, capital S, capital N, capital S, capital N, and capital N, capital N. Now we know exactly what would happen with these two right here. These are going to be a 50-50 box. With the capital S, that's going to be an individual that has that sickle shape no matter what. So this box right here will be all black. And this box right here will be completely normal, so this will be all blue, leaving you with this nice little pretty design here. And the phenotypic ratio would be very similar to what you've seen in the incomplete dominance, where we have a lot of the heterozygotes represented versus the normal and the sickle be smaller in number there. In this case, it's a 1 to 2 to 1 of normal, both, and sickle-shaped cells. All right, here's where things get really fun. You know that there are many, many, many different colors of eyes, which means that there has to be multiple alleles of eye color in the same spot for the gene that codes for eye color. In this case, we know that brown eyes occur more often than blue eyes, which occur more often than green eyes. So what if we just said, hey, seems like brown eyes are going to be dominant to blue eyes, and blue eyes might be more dominant than green eyes being the least dominant of all, almost recessive to the blue eye, which is recessive to the brown eye. It's almost like a scale, a ranking. And in this case, we will color the brown eye color with black markers, and we'll use a capital B for brown. We'll then color in the blue eye color phenotypes with blue marker, and we'll use a capital L for eye color. We'll just have to remember that the B is stronger than the L because we're using a capital L in there. The green colored pencil that we use will represent green eye color. That'll easily be masked by either of these two markers, of course, and the lowercase g will be our gene or a specific allele for that green eye color. So if an individual has brown eyes, then they have to have the dominant brown allele at least once. But the second allele could be any of the other letters, right? Because if I have a combination of capital B, that's automatically going to be brown eyes. If I have a capital B and capital L, we know that the capital B is actually dominant to the L, and therefore this masks that one there. And of course the capital B is dominant over the lowercase g, therefore we have brown eyes shown over the green there. In order for there to be blue eyes, it has to be a situation where the L is the dominant one over the G, or it's the only one available. If we combine that B and the L again, well, the B is still going to mask over the L, and therefore our only possible genotypes for blue eyes is going to be LL and LG. What does it mean about green? Well, it's the most recessive of all. Therefore, it is the one that has to be by itself and have its only homozygous recessive allele, that lowercase g. Okay? So this is a complicated section where we actually have now 6 different possible genotypes instead of the 3 that we normally have, because we added one more phenotype into the mix here. So let's do number 13 together here. Let's say I cross a homozygous blue-eyed parent with a capital B, lowercase g parent. We know that's going to be a brown-eyed parent, but they have that green recessive allele in the background. Okay? So, we're crossing a LL because it's homozygous. Blue-eyed is the L's there. It's going to be crossed with a capital B, lowercase g. And we're going to put the L's up here and the BG right here. If we drag the B's across here, that's the most dominant allele, so we might as well start with that. That'll be a capital B in that box and a capital B in that box. Then I'll go to the next dominant allele with the L's, so that would be a L next to the capital B there, an L next to the capital B there. Looks like there'd be an L in the front here and an L in the front there. And the lowercase g's would just fill in the rest of the boxes there, leaving us with 2 BLs and 2 LGs. The colors coding of this is going to be a little bit interesting here. Let's start off with any green alleles and color in the green first with that green colored pencil. So those lowercase g's are in these boxes, therefore we'll color in that box first all the way. Now we'll go back with a blue marker and color over any of these boxes that have a capital L in them, and looks like every single one will, so I color in all of it. Finally, we'll use the black marker to color in for brown eyes, and that would be these 2 boxes here that have the brown eye allele. So that'd be the black marker right there, giving you 2 black boxes and 2 blue boxes. Even though we know we colored this in green before, the green is being masked by the blue, and even though we know we colored this in blue before, the blue is being masked by the black. That gives us a phenotypic ratio of 2 brown-eyed to 2 blue-eyed individuals. Hence why individuals can lose their green eyes in a generation here. Or I should say families can lose their green eyes in a generation. Give number 14 a shot here. I'll pause the video and let you work through it. Remember that a green-eyed parent can only be the homozygous recessive alleles of G, G, so they're going to be the lowercase g's over there. And the BL parent is going to be labeled in this box over here. Everyone gets a lowercase g, but it looks like that lowercase g won't really matter in the long run because we have the capital B's and the capital L's to mask over them. So I'll color in all 4 squares with that green color pencil, but you know what's going to happen next. We're just going to color over half of them with blue and half of them with black, giving you another 2-to-2 ratio or 1-to-1 ratio of brown eyes to blue eyes. Even with that strong, or even with that recessive homozygous individual with the green eyes, looks like it's no match for an individual with blue and brown eyes here. Last one, last pause. Good luck. Let's try it out. So in this case, a BG parent and a BL parent are easy to put into the Punnett square here. You know you're going to drop down all the B's, drop across all the B's, and all the L's go right in next, followed by the G's that are most recessive. When putting this into our color coding one, remember we start off with the green one because the green one's going to be covered up pretty easily here. So these two boxes have the green allele there, so we'll put in the green colors there. Let's then color in the blue marker for blue eyes. That would be this box and this box here, covering up that G there. And finally, if we look at the black marker with brown eyes, we would cover up that box, and that box, because all three of those boxes have that most dominant B allele there. So this phenotypic ratio would be a 3-to-1 brown-eyed to blue-eyed, sorry about the typo there with the L, blue-eyed individuals, as in for every 4 kids, there should be 3 that are brown-eyed and 1 that's blue-eyed, and none that have the green eyes, even though they have the green alleles masked over. And that is it for the entire worksheet here. If you take a look, you've done every single one all the way down to the very end. But I have one challenging one left for you in this multiple allele here to see if we can get any green-eyed kids from any combination here, because Lord knows we do love some green-eyed individuals like me, right? So, if we take a look at the cross between a BG parent, which we know would be brown-eyed with the green allele being covered up, and a heterozygous blue-eyed parent, then we'd have an LG and a BG all connected together here for the cross. Go ahead and try to finish this out on your own. This will be some of the points that you get for your Ed puzzle here. So if we put the BG parent up here and the LG parent over here, then every single one of these boxes will have a B, every single one of these boxes will have an L, but it looks like all 3 of these will have G's, and this one right here has 2 of the G's, meaning it's homozygous recessive. If we look at it closely enough, we should be able to see right away that the green will be masked in these 2 boxes, but look at that. I haven't colored that one in yet with blue or black, so therefore that means that we have some green eyes in the mix here. The phenotypic ratio for this one would be 2 of the brown eyes, 1 blue eye, and 1 green eye. Finally, someone's having green-eyed children. Such a pretty color there. That is it for this Ed puzzle. Hopefully you followed along with the entire worksheet here so you don't have to do things twice. If you do, make sure you look at my slides or look at my answer key to take a look at which ones you might have gotten wrong along the way. Ask questions. Feel free to stop the video and come back to another time. That's it for this Ed puzzle. Let's move on to the next thing.